like for example, odorants that fit the receptor, like think about like isotopes, they'll all fit the same receptor, but certain isotopes smell different, right? Like for example, some people say drinking water obviously is flavorless, but then you drink deuterium water, which is just hydrogen with another neutron on it, then it tastes sweet, right? Even though they're the same key, but they taste different, right? So maybe the vibration theory can explain that for us better. So, but then vibration theory also doesn't get it right because some molecules have different vibrations, but then they still smell the same. So perhaps a good theory is like a mix of the two. So I thought this was super interesting, changes the way we think about how we smell odors. But yeah, now we'll skip ahead to slide 139. Okay. So yeah, let's go straight into the brain, this idea about the quantum brain. But just before the quantum brain, so these are some brain questions like, what is consciousness? Where do you think our memories are stored? Last time we might have suggested that the memory is stored in the microtubules, in the centrioles of our neuron cells. What's the computational brain power of our brain? I remember as a kid, some people said we are utilizing 10% of our brain's power. Is information processing in the brain, is it classical or is it quantum? And like considering we just have to eat like donuts and stuff and we can power our amazing brains, whilst like today's like Google supercomputers use like kilowatts and kilowatts of energy. So like how are we able to do so much at such a low level of power? Right. So there's a quote by Robert Epstein, who said like, our brain does not process information, retrieve, or store memories. In short, our brain is not a computer. So what he was saying was that it's like comparing apples and oranges. Like our brains aren't even a computer, so you're trying to fit the computer model onto the brain. It doesn't really make sense. And so we give some examples, like like brain scientists, they're never gonna find a copy of that music that's stuck in our head, right? It's not like located right here, or like where did we remember the word fantastic? It's not going to be right here. So he's saying, of course, like our brain's not empty, but it's just that our memories aren't so localized as like memory on a computer would be. Okay, so let's go through like where's the memory stored, where's our computational power coming from. So we know there's 10 to the 11 neurons and 10 to the 15 synapses, and they're operating like at 10 hertz, right? 10 impulses a second. So wow, 10 to the 16 synapse operations per second. And a pretty, not, who knows if it's relevant, who knows there's correlation, but the blue gene, which is a supercomputer, performs 10 to the 15 operations. So it looks like we have a magnitude of 10 more operations per second than the fastest supercomputer. But then ironically, our brain only uses 25 watts. I mean, I think there's some light bulbs that use 25 watts, right? And then blue gene requires like 1.5 million watts. So big difference. So the question now is like, is quantum mechanics relevant to the brain? Is our brain a quantum computer? That's why like classical computing can't really explain it. So, do you remember we were talking about the wave function where like an object can be yes or no, or not, sorry, yeah, like we can't know an object's position or an object's momentum at the same time. You can only pick one. So some people think our consciousness is always in a wave function. It's in a superposition wave function, right? We don't know its position or its speed at the same time. But the moment we have a thought, it collapsed, and now it's definite. We either know its position or we know its velocity or momentum. So some people think that's what our thought is. And then like, are there quantum limits on how much we can predict about the brain. Like intrinsically, quantum mechanics is just probabilistic and very hard to understand currently. So does that mean we can never really predict the brain? And then how does it imply for artificial intelligence, right? Like right now, all our AI is not quantum. All our AI is on classical computing. So is like quantum mechanics and quantum computing the gateway to a next level of AI? Okay, so the brain's functional unit is a neuron. So I guess we'll go into like some details about a neuron. Yeah, so looking here, that tube is that microtubule we talked about last time, the microtubule. And microtubules, again, made of two things. I believe it's alpha beta tubulin. So last time it was suggested that the exact arrangement between alpha beta tubulin leads to our memories, right? But in this case, there's a model here that's suggesting these alpha beta tubulins are actually like qubits. And in quantum computing, qubits are like, they exist like either spinning up or spinning down. It's just like either it's up or down, like Schrodinger's cat, either alive or dead. So constantly existing in a alive or dead form. In this case, it's constantly existing in a blue and red form. And then the moment our neuron makes a choice, maybe it collapses into one form. This is just one theory here. Right, so here, this model is just showing like the electrons either at the top or the bottom, and this two conformation of tubulin is like, it's happening at the same time. And then once we have a realized thought, it just picks one tubulin. So this might be a way to describe our brain. So let's talk about the energy costs here. So again, 10 to 11 neurons. So now we're saying it's 10 to 18 operations per second. That's the brain, that's the whole entire brain. And then within a single neuron, there's 10 million microtubule tubulin dimers, right? So, they're constantly switching around. Like each, there's 10 million tubulins, and each of them are in a superposition, switching back and forth between up and down, or blue and red, as the previous slide showed. So that would lead to a potential 10 to the 27 operations per second in the brain, using this idea about tubulin switching back and forth. So previously you said it was probably 10 to 18, but if we use this quantum qubit theory back here, like this tubulin switching between blue and red form, then we get 10 to the 27 instead. So, how can this be energetically sustained? So now we look at some examples of what 25 watts gets us, right? So what does 25 watt gets us? Obviously it gets the light bulb. What else? So in our cells, like 25 watts is like, of that, 60% is used for ribosome protein synthesis. So we think that, a large proportion of our body's energy is just making proteins. Makes sense, right? Like the DNA transcription, making proteins. And then apparently 70% of the rest, so 40%, 70% of the 40% is to maintain our core temperature. So that means that we only have 5 watts left, right? So we only have 5 watts left. And so basically we only have 5 watts to process at 10 to the 27 cycles per second. Minimum energy cost. So, in physics, like, especially in quantum mechanics, quantum physics, they always want to get down to the lowest possible denominator, because it's not an infinite gradient. It's always like quantized, right? So here they quantized the lowest possible increment is 3 times 10 to the negative 21 joules. That's the minimum amount of energy you can have to have some information. 1 bit is like the lowest amount of information possible. In biology, obviously, our unit of energy is not, or our unit of energy is not a joule, it's a ATP, right? So ATP uses 5 times 10 to the negative 20. I mean, the basic key is like ATP is 10 times more energy, right? So here it's saying at most 10 to the 20 bits per second. So that's a theoretical, just based on the energy, right? Just based on the energy, we should only be doing 10 to the 20 bits per second, but we're doing 10 to the 27. So that's what some people are saying. That's just about information processed. So I guess let's look at some time scales here. So 1 nanosecond for microtubule, and then 1 microsecond for tubulin conformation changes, 1 millisecond fraction potentials, 1 second for brain pre-processing times. So I think the key is like the second one, the 1 microsecond for tubulin conformation. That's the one where the tubulin's existing as a red and blue state at once. And once it collapses, apparently our neuron makes a decision. But it's interesting to contrast that with the 1 millisecond action potential. That's the one everyone's most common with, like how our nerves just conduct, right? How fast is that? It just takes 1 millisecond for the ion channels to open or close, right? Very fast. And this tubulin is just 1,000 times faster, right? 1 microsecond. So still less than 1 watt use to cognition. The other stuff is just trying to keep us warm and making proteins. So the amount of information processed depends on the clocking rate, but that must be less than 1 bit per neuron per nanosecond. So it looks like this is like that quantized lowest increment level that our clock speed of our brain can be. So let's see, if 1 nanosecond for a microtubule excitons, then all microtubules in a neuron must be coherent. So there's 1 bit per neurosecond. Okay, this looks like some intense math. So I think we're gonna skip it. Right, the memory storage. I guess we'll go in a bit more depth here. So, everyone has a USB stick probably these days. Maybe not. I mean, Apple's taking away those USB ports, but they've gone into like 8 gigabyte, 16 gigabyte, right? They hold some of our homework, some videos maybe. So let's see how we can convert that gigabyte idea onto our brain's neurons. So obviously back to our microtubules, the tubulin, there's alpha beta. And if you're an alpha beta, you have 2 conformations. If you're beta, you still have 2 conformations. So we're just seeing all the combinations possible that could lead us to have that memory, right? So we're saying for phosphorylation states, there's 4 per dimer. So each dimer can already have 2 conformations, and then each conformation can then have 4 phosphorylation states. Okay, so we're adding up and it just becomes 128 states per dimer. That's how the math works out. So just a single dimer, 128 states, right? That's like a lot. So if we combine that together, it comes out to 1 gigabyte per neuron. I mean, how crazy is that? What a coincidence that apparently 1 neuron is 1 gigabyte. And then I guess the easiest metric would be just counting how many neurons we have in the brain. And on average, 100 billion neurons. So that'd be 100 billion gigabytes. So that's a lot. Wow, 100 billion gigabytes. And then they're just again comparing it to the supercomputer, which has 10 to the 15 flops, and then our brain has 10 to the 26 flops. So that is like 26 minus 15, that would be 11, 10 to 11 more flops. That's crazy. That is absolutely crazy. Would that be, that's 6 is a million. Wow, I don't even know what you would call that. That's crazy. So on to the next one. So yeah, what is memory? Unlike computer memory, like our memory goes away, right? When it's on a computer, if there's no disturbances, it's on there forever. So, our memory unfortunately is not quite like that. So obviously it's very different. So we can encode, store, sleep helps with encoding, storing the memory. Then we have to recall it. My friend, I think, took like it must have been a psychology course. His prof said a really interesting thing that I didn't understand at first. It was a simple quote. He just said, remembering is forgetting. And I was like, what does that mean? Is that like a riddle? But I guess it's true. Like if we remember something, if we remember it, then times that we forgot it and then we remembered it again. So I don't know if that's what I meant, but he just left me with that quote and I thought it was pretty interesting. But yeah, back to this slide. So it's postulated that it's not like local, our memories, but it's more like a network and it's just interspersed along these networks in our brain. And like plasticity and long-term potentiation, I think are very important concepts just to know in general what they mean. So both of them just tell us that's why we can adapt, right? That's why we can change our attitudes. We used to bite nails, maybe we don't anymore, right? We broke habits, we maybe added habits. All of that is because of our synaptic plasticity and long-term potentiation. So what do these words mean, right? All it means is, at the end of this neuron and onto this neuron, there's a synapse, right? And let's say if I want to bite my nails, I used to have lots of biting nail receptors that get excited when I bite my nails, right? So a long-term potentiation example would be, I want to break this habit. So now my synapses just lost. Before I had 100 nail biting receptors, now I only have like 12. So now when I shoot a nail biting signal, it doesn't excite this one that much, right? Before there was 100, so just one nail biting signal would excite this one so much, and I'd want to bite my nails. But because long-term potentiation and synaptic plasticity, this is a synapse right here, and it's plastic. That means that we can change our habits, which is basically changing the number of receptors there, and that leads to different behaviors or different, maybe we remember new things. You know what I'm saying? It's a very simplistic overview, but that's what long-term potentiation means, just changing the number of receptors to a specific response. So now that we know like how the brain kind of works, let's dive in here. Okay, so so this guy's saying, he hypothesized 3 times 10 to the 20 bits. Total life experience. Okay. Oh, this is just showing like people have different hypotheses for how many bits we can store. So very interesting. But let's look at Launder, the very bottom one, the 1986 one. This is very interesting. So he said 10 to the 9 bits. So let's say we retain 2 bits per second of visual, verbal, and music memory. Every second we retain that. That would mean like if we have 10 to the 9 bits of storage, we have 2.5 billion seconds. So that's our lifetime. That's interesting. That would be a way to measure it. Obviously, it's a big assumption here that we're recording at 2 bits per second. Who knows? I'm sure in real life, sometimes we just like completely turn off and recording 0 bits per second. And it's something we might spike it and record 100 bits per second. Maybe it's more like that. Let's see. Cytoskeletal involvements. Yeah, so again, probably microtubules. Yep, so microtubules again. And I think in science, another good thing scientists try to do is keep everything the same, keep everything constant, and just change one thing, right? That's how you might be able to get to a causal inference. So here, how scientists might come to the conclusion that microtubules are responsible for our memory is that certain microtubule diseases in our brain actually cause memory loss, right? So that is a big implication that certainly microtubules are somehow involved in memory loss, or it could be the other way around. It could be that our memory loss was caused by something else, and then that caused microtubules disease, right? Who knows? But this still gets us closer to answering how microtubules are involved with our memory. So CaMKII 2 is just like is that Calmodulin kinase. I'm not really familiar with this one, but let's see what it does. It looks like it affects the receptors here, right? So it looks like this thing is CaMKII 2, and somehow it's involved receptors. And again, receptors are again involved with long-term potentiation, right? More receptors is more encouraged, right? And I think we'll just skip to the next slide because this is very cell bio. Right, so here we see, right, LTP, long-term potentiation. In this case, we have more receptors, right? It can also go the other way around. That would be long-term, I think it's called long-term depression. LTD maybe, something similar to that. I might be getting it wrong right now, but it can go the opposite direction and it can suppress and be long-term depression, I think. So in this case, long-term potentiation, which means more receptors there. And so that's at the cell level, but then perhaps at the molecular level, that's more microtubule phosphorylation. Oh, I see. So CaMKII 2 was involved with the microtubules And kinase always means phosphorylation. So, kinase means it's either putting a P on a molecule or taking away a P, right? So in this case, it's putting on a P, and that might change our microtubule properties. I think in a previous slide, it said that each dimer has 4 phosphorylation versions of itself, right? So that means that this CaMKII can change a dimer, like almost put 4 different pieces of clothing on it, right? And see how that all those combinations lead to our memory. Okay, so wow, so it looks like this kinase is vital for a memory, right? And vital for a memory. I think in a previous slide it might have mentioned also that certain diseases with this kinase. Imagine you had a mutation in this protein, right? This Calmodulin kinase 2. Then it looks like it implicates that we have memory impairment and stuff like that. So yeah, very interesting. And at the same time, it might also imply that we need calcium to have good memory, right? Because this big protein, this CaMKII, only works with the presence of calcium influx. So that's also very interesting. Okay. I don't know if I understand this one too well. Field lines convergent, showing electrostatic attraction. Considerable binding energy. Well, and I'm not sure exactly what this slide is talking about, but it just looks like CaMKII has some affinity, a stronger affinity for something. Maybe it has a stronger affinity for a microtubule. I'm not sure. So we'll skip ahead. Okay. So here again, I think previous slide said we have 4 phosphorylation states that a tubulin could be. So again, reiterating here that it looks like we could get up to 1 gigabyte per neuron based on all these bits, different states. And the first like 4 points here, this is still classical computing, and then throwing in at the very end, a quantum computing, and this is a completely different concept, right? Like all this work, we're still talking our brain as if it's a normal computer, right? Not even a quantum computer yet. So, okay. So again, phosphorylation sites, I think this is just, we can appreciate how many different combinations the same tubulins could be, right? This is the same tubulins, and that CaMKII, was it CaMKII? Yes, CaMKII coming in and just putting P's on different tubulins and completely changing the combinations, right? It's like we have, was this 4, that's 8, that's 5 plus 8 is, what is that, 13. So we have 13 tubulins here, and just look at all the different ways these 13 tubulins can be arranged just from one enzyme, the CaMKII, right? Changing the phosphorylation states. That just adds so much different combinations. So they're suggesting 10 to 20 bits per brain. 20 minutes per brain. Okay. And each event costs 10 KT. I wonder what unit KT is. Is that a unit of energy? KT? Not sure. Let's see. Okay, so this is a paper. These represents a paper. So this paper is suggesting phosphorylations again. So that CaMKII phosphorylating serine, which is amino acid, the C terminus of this serine and of the microtubule, and that is possibly how our memory is. Very interesting. Also, now we're talking about the mechanism of how this phosphorylation would lead us to have different memories. So it looks like these phosphorylations, depending on where it is, right? It could be up to 4 states. That kind of tells MAPS, which is another enzyme, where to bind. And then telling that where to bind changes neuron structure, transport, synapse structure. And I guess the combination of all those things dictates our memory. Wow, it's very interesting. So, and or are like these are computer science terms, and specifically these are like hardware computer science terms. So, you know, on our computers are so they're just combinations of and or gates, right? That the gates control can electrically flow or not, right? If this and this is true, like this and this, then the electricity flows together, or is something else. So I think they're suggesting here that our microtubules are literally like our computers, chips, and in our phones and computers that follow this logic, right? Depending on their phosphorylation state, it dictates what kind of and or gate they are. Okay. Let's see what this one's talking about. Few fast, so very few but fast transitions, but many processing units must be correlated. Many slow, uncorrelated per brain. Okay, so it's saying for this hierarchical model to be true, there must be very few but fast transitions, but then have many processing units. We have 10 to the 18 tubulins in a brain. But I think in real life, what we see is there's a lot of slow transitions and very few. There's many slow transitions, but few uncorrelated processing units. So I think this paper is saying the bottom one is the real life. The top one is what the top one is what our model is based on. So I think basically it's saying that our brain's not a classical computer, right? The top is explaining our brain as a classical computer, and the bottom is saying the brain is completely not a classical computer. What do we do now? And I think this is where quantum computing comes in. So perhaps the only way we can truly talk about our brain the most accurately is through quantum computing. So quantum computers use entanglement and coherence. Okay, so now we're getting to a biological explanation. Again, very like, I don't think this is tested, but this is just getting into the nitty gritty a little bit. So delocalized trip, so tryptophan excitations could serve as a basis. Okay, so applying different quantum ideas. But onto quantum entanglement coherence, just for the first point over there. So, you know, what entanglement is somehow, I talked about last lecture, but somehow two entities can be entangled and then they'll behave identically. Like if this one jumps too, right? Obviously they don't jump, but that's the idea, that they're entangled. And it doesn't matter the distance between them. They could be like a light year away. They're still theoretically entangled. So, I mean, that is very interesting to me. And coherence. So coherence is just, again, sometimes up, sometimes down. But if we take a zoom out look, somehow they might be coherent, right? And the moment they're coherent, they have information. So most of the time, discoherent, you know, up and down, but then they can be pushed into states of coherence, and then we get them to do things that we want them to do. So that's quantum coherence. Okay, so again, destabilized microtubules. So again, we're wondering how, you know, our brain has lots of memory problems, and it seems that when we destabilize microtubules, we have memory issues. But again, so in this case, we're suggesting that it's a trigger. So it's the cause, right? It's a trigger, it's the cause. And the moment it's triggered and caused, you know, what are the consequences, right? And so we're implicating here that microtubules alone are likely causing, you know, Alzheimer's, Parkinson's, Huntington. The first three, by the way, are like, we have no cures. Like, no humans haven't made the cures for this one yet. And then some brain injury, and then many other diseases that aren't even brain related. I mean, we have heart and cancer disease. So wow, that's very interesting. So not only correlated, perhaps might even be the trigger to some neurodegenerative diseases. So looking at Alzheimer's disease, you know, beta amyloid plaques, that's a classic right there. Right, so you have beta amyloid plaques and tau. So I think these are the two to know about. whenever you hear about Alzheimer's disease, you'll always hear about beta amyloid plaques with them, right? And I think currently scientists, cell biologists, they're still debating, is beta amyloid plaques a consequence of Alzheimer's disease, or does beta amyloid plaques cause Alzheimer's disease, right? That's a big difference, right? It's just like, if a car's leaking oil, that'd be beta amyloid plaques. And are you assuming the leaking oil is what's causing the Alzheimer's, or is there an internal problem with the engine that's causing an oil leak, right? So that's still a point of debate right now. And so it looks like beta amyloid plaques are correlated with cell death and neurofibrillary tangles, memory impairment. Also very interesting about this beta amyloid plaque. Normally, in our cell, it's all liquid, right? It's an aqueous solution, and all our proteins are dissolved in it, just like we put sugar and salt inside our water. We can't see them, they're all dissolved. But what's interesting is beta amyloid plaques are insolvent, so they like come out of solution. So imagine a cell inside, it's just all fluid, it's all full of proteins, but it's fluid. All of a sudden we have things that are not being dissolved in water. So they're literally like physical things not dissolved in water. So that could definitely lead to problems. Ooh, so what comes first, right? Is it tangle formation or microtubule deterioration? So they're saying likely microtubule deterioration is a primary event before the tau proteins clump with the neurofibrillary tangles. Interesting. So tangles form where microtubules disintegrate. Okay, so that might be a reason why those tangles form after the microtubules disintegrate. Interesting. So there's a labile domain and stable domain. So not sure how this necessarily relates to the idea of like quantum computing, but the microtubule, remember, it's a very long train track. The stable domains are like concrete, they don't really move, and the labile domains are like constantly active. They could shrink, expand, because the microtubule needs that. Imagine you're like an amoeba, that big cell that globs around. If it was all stable, it'd take too long for it to glob. So certain regions have to be movable, so then it can extend its cytoplasm forward using the labile domain of the microtubules, right? So looking here, it looks like the top is when there's tau, the bottom no tau, and the tau is the blue. So when there's no blue, the labile region is gone, but the stable region is still there. And then after MAP6 depletion, the tau comes back. Okay, very interesting. Okay, so spine loss. Wonder what spine are talking about here. Microtubule spines, or is it the neuron spines? Okay, but is it getting pretty advanced here? But I think it's still talking about the same idea here, right? So you have this destabilized microtubules, and tau is supposed to maintain the microtubules that are supposed to constantly change states. I don't really want to talk about this, to be honest. I think this is like super cell biology. I'm a cell bio major, and I don't know if this is super. I don't want, if we go too in depth, we're gonna lose the flow, of the quantum computing here, because this is like completely not quantum computing anymore. So let's leave this cell bio bit. Oh, I guess this is kind of interesting. So it looks like, this first state is an example of short-term potentiation. This is our short-term memory, like just memorize 3 words before like an English quiz or something, and then permanent storage. So it just looks like our microtubule networks are a bit more structured and organized in our long-term storage, whereas our short-term storage kind of organizes itself, but not to the degree of organization for a long-term storage. Interesting. So why do microtubules deteriorate and initiate Alzheimer's, right? So we know the biggest correlation when it comes to Alzheimer's is age. Age seems to be the #1 biggest correlator. Okay, so with age, we have changes in gene expression. 4% of genes in the brain show increase or decrease. Interesting. So 4% of our genes in our brain start changing as we get older. decrease in synaptic protein, right? So decrease in synaptic protein, that right there is LTD, right? Long-term depression, right? That's the opposite of potentiation, which is increasing those receptors. Now we're decreasing it. Decreasing cytoskeletal protein, so that's just losing microtubules right there. And then just increase bad things, right? Inflammation, stress. So with age, genes change in our brain to make it a worse environment for our neurons to live. So not great. Anesthetics, right? I think there's a concept of quantum anesthetics. So I think we're going to get into that right now. Let's see. So let's see how microtubules are related to anesthetics here. Bind specifically to tubulin in brain in humans. Taxane-treated patients are resistant to anesthetics as destabilized microtubules. Oh, wow. So I think just looking at the underlying word is what matters here, is that microtubules seem to also interact with anesthetic molecules. So hence it's possible that neuronal microtubules not only store memories, but can also be location for conscious processes, right? If it's a location for a conscious process, when there's anesthetics, it destabilizes the microtubules. So, and it anesthetizes us, right? So that might suggest that our microtubules are also a place of consciousness. Okay. So it looks like the way anesthetics work on our microtubules, the tubulin, is it just changes tryptophan excitation. And tryptophan is excited just by phosphorylation. You put a P on a tryptophan, it's excited. So that's what we're talking about here. Anesthetics disrupt resonant energy transfer in microtubules. Yeah, this is advanced, but I think looking at words like coherent, you know, that hints a little bit quantum, oscillating, and then a big math equation. Geez. So, right, anesthetics possibly disrupt quantum processes taking place in microtubules with close similarity to energy transfer process taking place in photosynthetic complexes. So relating photosynthesis to this is a good way because we know photosynthesis involves quantum behavior. So if scientists are seeing a similar energy transfer process seen in photosynthesis, which is quantum, perhaps the energy transfer in our microtubules is a quantum event. Okay, so here we have a bunch of anesthetics. I know, like, you know, when scientists are putting rats down, they use, I think, nitrous oxide or something for the rats. So let's see. Oh, wow, this paper was by Tazinski himself. So this relationship correlates very closely with the potency of anesthetic molecules that affects our brain. Yeah, so just look at the right graph here is just showing the potency of different anesthetics. So 100 is this y-axis is the minimum amount needed, right, to be an anesthetic. So 100 means it's not very strong. So nitrogen, you know, we even breathe that in the air, right? So somehow it's a little bit of an anesthetic, but you need a lot, right? 100. We go down here. So these ones are the chloroform. Right? Isn't that what people put on the cloth before they like subdue someone? So right, these are very strong anesthetics. So, you know, this trend is almost the same trend here, where we see the y-axis is now the frequency shift. So frequency shift is using frequency shift, it actually predicts the amount of concentration needed to kill someone. So nitrogen has a very high frequency, very low frequency, whereas like a chloroform down here might have a high frequency. I don't think it's important to memorize if it's high or low frequency. It's just interesting to note that something like how potent anesthetic is correlates with its different frequency. And frequency is just a completely different measure, right? So it's just saying how are two completely unrelated things also correlating the same thing, right? I think that's what's interesting here. Alzheimer's disease, I will skip this slide. Okay. Brain bandwidth, 10 to 30 hertz. This slide is, wow, this is, I don't know what's going on. So focusing on the dendrite. Okay, so dendrite is just the beginning of a neuron. So the neuron has a beginning, which is a dendrite, which is like a tree, just so many branches, right? And each branch is a synapse. So each branch is like a cleft. So the beginning of neuron is just a bunch of clefts full of a bunch of trees and branches, and it becomes a long, thin noodle to become the end. So we're looking at the dendrite here. Makes sense, right? So again, dendrite have those little clefts and they interact with all the other neurons. So here we see, you know, another neuron there, another neuron there, interacting with this huge branch of a dendrite. Okay. Previous and current study. So in previous, highlighted in red here, is just talking about, you know, previously we're looking at those microtubules inside here, but maybe now let's focus instead on the dendrite or the interaction with the receptors and the previous neuron. Okay, again, nothing new here, just saying that neurodegenerative diseases, you know, correlate with deteriorating cytoskeleton. Obviously, microtubules are a part of our cytoskeleton. Alzheimer's disease, same thing. Very biology, to be honest, very, very biology. But Huntington's just basically another very similar thing to Alzheimer's, completely different symptoms, but the same idea where there's an alpha-synuclein and parkin that, you know, change and become insolvent, and then bad things happen. So same thing. Okay, so we have just all these diseases. So we have traumatic brain injury, epilepsy, ALS. All these are nerve-related disorders, and obviously when there's nerves, there's neurons, and all of them are somehow involved with cytoskeleton. So I think the main takeaway here is that cytoskeleton is involved with neuron-related diseases. Memory storage. Yeah, so one way is like memories are not stored in the brain at all. Okay, so that's one way to put it. And it looks like there's been some studies that show that even a plant can learn. So again, a hard drive or SSD, the things that are in our computers, they store things at one location, right? It's like at hangar 2, put this information in. But again, our brain doesn't really store it so locally. It seems like it's either a network or not stored at all. So let's see. So is memory localized? So we're testing, is memory just like a hard drive? It's just right here. So this is some kind of worm, and we decapitated it. So that's straight up a loss of localization, right? It's like we cut off a piece of the brain, but then the head regenerates and this worm apparently retained its long-term memory. So any memory in the brain, if it was localized, would have been gone, right? If the apple was right here, this worm would never know an apple, but it regenerated and it still knew what an apple was. So that suggests that memory isn't really localized for an animal. Memory, intuition, gut feeling, right? So this is a very interesting point. You know, we all have the gut feeling when we see someone or we're in a situation, and we instantly have a feeling, good, bad, excited, sad, right? And you know, some people think that's an emotional thing, but perhaps that's really our subconscious brain doing thousands of logical assumptions and experiments, and instantly coming to a logical response. But that's reflected subconsciously. So we never got to experience those thoughts because it was subconscious. But in the end, it's reflected by our gut feeling. Do we feel good or bad about a situation? So, you know, before often, I think at least, I feel like in previous generations, it was like encouraged to go off gut feeling. I think these days it's not so encouraged, but perhaps it should be encouraged, right? Maybe gut feelings represent logical thinking, but just subconscious. We just don't get to, go through those thoughts, but our brain does it for us, right? So maybe we should trust it. Who knows? And can we measure this ability? Can we measure intuition or gut feeling? So obviously, these down to just non-invasive ways, like the lie detector, for example. It changes, lie detector just function that changes in the slightest like sweat, right? So let's see. Historic perspective. Okay, so human 1 to 19 bytes. Number of words ever written, number of words ever spoken. We have this many bytes. Okay. Data on a digital media, 3 times 10 to the 19. Okay, so we're just getting some basic numbers here. Again, just a microtubule store information, so we'll just skip this slide immediately. These are just some repeated slides, interesting. So we went over this already. Okay. Kinase regions found to closely match microtubules. Oh, okay. So even though we have the same number of microtubules here, I think, just changing their overall structure makes them, for example, this one is more likely to bind to that CaMKII, right? Because it's in that shape, whereas these ones maybe not as likely. So I think it's just saying that even geometric matching might encourage a CaMKII to interact with a microtubule. Ooh, so now we're going on to like those anesthetic drugs. So yeah, I believe normally it's grouped in these 4 things. We have stimulants, depressants. So obviously alcohol would be a depressant. Stimulate, would that be like cocaine or something? We have caffeine here, right? Cocaine, hallucinogens, probably LSD, something like that. Antipsychotics, are these opioids or CBD? That's marijuana. Okay, this is an interesting slide. Computational predictions. What's happening here? Yeah, I think what's important is just this last line here, that again, we think our microtubules are involved in some quantum things, right? Because they could be blue or red at the same time, that's quantum. And then when we anesthetize a microtubule, we lose conscious thought. So maybe there's quantum things happening in the microtubule. So I think overall it's just saying that compounds like the anesthetics interact with quantum information processing in the microtubule. Common drugs, right? So we have common drugs here. It's always cool to look at their shapes, I think. Right, like alcohol is such a simple compound, it does so much. So interesting. And also, like chocolate and caffeine they look similar, right? Like what's different here? Wait, are they the same thing? Oh no, they're not. There's a carbon group, there's CH3, and there's only, wow, I didn't know that. So caffeine and chocolate are just off by 1 molecule. That's crazy. Adrenaline makes excited. I wonder if it correlates, like, does chocolate make us awake? Is it caffeine? Who knows? Let's see. Nicotine. Yeah, very interesting. Okay. So previously we were talking about memory, which some people think might be easier to explain. Now we're on to like consciousness, right? Much harder to define. Let's see. Again, just talking about quantum computing, once cold and isolated, but our brains are so warm and mushy, right? So how are they kind of similar? Okay, so we're moving on. These are points we already talked about. Let's read the hypothesis out loud. The microtubule network in the dendrites at the front of the neuron is related to memory, and interaction with anesthetics can influence consciousness and alter memory formation. I think just the bottom part alone makes sense, right? Some anesthetics we take obviously influence our consciousness, and obviously alter our memory. But I think the hypothesis here is just connecting the microtubule part to what we already know. We already know that certain anesthetics, given in the hospital, can alter our memory, alter our consciousness, but it's just like microtubule networks interact with anesthetics, and that's why this happens. So that is the main point here. Okay. General anesthetics possess dissimilar structures, of course. So just a range of structures. Though quite honestly, they look much simpler still to those previous drugs like caffeine. So very interesting. I think cyclopropane, very interesting. Like it's a 3-membered ring. Forgot, is that stable or not stable? Very interesting. Chloroform. Okay. Intravenous. This is inhaled. Oh, makes sense. I guess inhaled things would have to be smaller molecular weight, so they actually can be in the air. And intravenous, obviously, probably a liquid form now. Okay. Propofol, milk of amnesia, famous for killing Michael Jackson. Okay, so that's cool to know. This drug killed Michael Jackson. So what about anesthetics? There's analgesics, hypnosis, paralysis, amnesia. Let's see. Okay, so volatile anesthetics reduce polymerized microtubules. So polymerized means the microtubules used to be a train, and these volatile are the inhaled ones, these small ones, reduce the trains of microtubules. They're just breaking apart these microtubules. So the point here is all these anesthetics, all the ones listed here, somehow affect tubulin. Sorry, I'll go back. Somehow affect tubulin or the microtubule itself, and they're all just interacting with microtubules. And the fact that we know that these drugs cause the effects that they do, and zooming in, they affect tubulin, somehow the microtubules and tubulin must be involved with like our consciousness, right? So I think that's the connection that's being made here. Okay, so there's 47 sites, so many dots here, 47 sites for binding of different things. This is just an alpha beta tubulin, alpha, beta, and so many sites here, okay. Matter perception, quantum psychology. So we're moving on. So that was, I think, quantum consciousness. We're going on to more quantum physics. We're almost done this lecture. Quantum physics, okay. Reality is probabilistic and uncertain, okay. The universe is a cloud rather than a clock. Possibly including our mind and brain. Okay, okay. I think this is a really important point. This is like a really philosophical thing, that there's no single objective reality. How do we know that we see the same red? How do we know that we see the same blue? How do we know that 1 hour feels the same for everyone? That science right now can't explain, right? So right off the bat, we don't have a single objective reality that we can share between us. We just have to believe we all see the same thing, right? We call this wavelength red, and hopefully we all see the same color, right? At least we know it's red because we're all seeing the same wavelength, but we don't know if we truly feel what red feels like, right? So that's the first step. We don't have that single objective reality. The current definition of such psychiatric phenomenon as delusions may hold. Okay, so current concepts of delusion, so beliefs held with conviction, which are impossible or false, rely on the assumption that one single objective reality exists, right? So sometimes friends might call each other, oh, you're delusional, but maybe in their reality it's true, or maybe in your reality that's truly your reality. So are they delusional? Delusional relies that all of us experience the same thing, like all our realities are the same. And interesting, Wernicke said that. I think there's a region in the brain for Wernicke. I think it controls our comprehension of words, I believe. Controls, like, can we understand what's being said? So very, very philosophical point, actually. So maybe our reality is subjective. or there's a missing link between what makes reality real, or reality is real, but there is more than one version. Very philosophical points. So, given how subjective psychology can be, like, and philosophical, right? Philosophical and subjective basically means there's many different opinions. That's where classical things don't really fit in. Classical things are very determined, right? I push this table down, it feels my push, right? Quantum isn't like that. Quantum is all about, a bit mysterious, a bit like superpositions, everything happening at once. So, I feel like every time scientists might feel like something can't be explained classically because there's just too many versions, that's maybe you bring in the quantum and see if this model might fit better. Like just mental states alone, right? Let's see. In mental states, the processes that either spontaneously or external interventions can make us excited or more towards depression. Yeah, that's very true. Like our emotions are affected by other people around us. If they're excited, we might be excited. So very interesting. Let's talk towards quantum psychology. So in red are the things that we put a definition to, and in black is just how quantum mechanics might be able to explain those things. So like, let's say multiple personality disorders, okay. So maybe that's just a transition between fermions. I don't really know what these are, but just a quantum mechanical thing. So these are all like potential reasons for what we see. Okay, like schizophrenia might be just Q deformed operators. Don't know what that means, but the point is, psychology very hard to explain classically. Maybe quantum theories might be able to explain it better. Meditation, right? I think many people, I don't think there's anyone that really says meditation led to a bad result, right? I think most people think meditation feels great. And maybe it's because their brain, is a quantum-like, and it's always discoherent with all these different thoughts. I find that too, like, sometimes you just try to think of nothing, which is what meditation is, trying to just empty the brain, think of nothing. It's so hard. The thought always just jumps back in. Maybe it's so discoherent, right? So maybe meditation helps make us a coherent state. So very interesting. So same slide as last slide. Quantum psychopathology. Okay, so pathology is talking about the physical anatomy of how this quantum psychology might fit in here. So these are just opposites. Either you're psychotic or not psychotic. I don't know what this one means. Impulsive or controlled, anxious, not anxious, autonomous or enmeshed. Okay. Interesting. So transition states just means like it's not just yes or no. It's not like we're pregnant or not pregnant, right? It's not we're normal or we have illness. Perhaps it's a transitional gradient. And that's why sometimes even though we're quote unquote healthy, maybe we experience some psychiatric problems temporarily, right? It's a graded severity of illness. Yeah, okay. So what are some future goals? Right, always just trying to make a model, right? In this case, an explanatory framework to explain all the phenomena we see. Quantum statistics is actually a risk assessment. Okay. Oh, I think we're talking about quantum embryology now. Okay. So I guess it's more of a philosophical thing, but how did this round thing become this? Or again, how did our DNA information become a turtle, right? Same idea. Not an expert on embryology, if I'm honest, so we'll just, I guess, enjoy these pretty pictures for now. But yeah, these cleavage events, I think we're a little bit familiar with that. And all these cleavage events are determined, I think, by the DNA. So DNA telling when to cleave, where to cleave. So what was this one? Exile development, still exile development, just changing. Interesting heat timing at 20 degrees. So it looks like changing temperature changes the timing of all this. I'm guessing maybe a higher temperature either destroys this process or a higher temperature might make this process faster. Very interesting. Things also still developing. No increase in dry rate since it was an egg. That's a really good point. So like going back to the original egg, the weight's the same, which is really important, right? Like let's say this was like 1 gram. At the very end of it, it's so complex, it's still 1 gram. That's extraordinary. Cell say splinter. So these three are just part of the cytoskeleton, right? Microfilaments. Okay. Cell state splitter. Okay. Yeah, this one, literally no idea what's happening here. This is looks very advanced. It looks like they somehow managed to fit it onto this curve. Differentiation tree. Right. I mean, the point here is like our cells are able to just follow the same tree every time, right? So like how is it able to launch these events at the right moment at the right time? And how does it know when cleaving is done, right? How do you know when no more cleaving? So just unsolved problems, unexplainable problems right now. So Conrad's Waddington's epigenetic landscape. I think that might be a model that this scientist made, and somehow it sees patterns all throughout nature. So in this case, the patterns seem to fit the embryologic, the differentiation tree, right? It's the same idea. Like there's that golden ratio, those spirals, and we see those spirals in like shells or like the way like flower petals are arranged. So in this case, Conrad's Waddington's epigenetic landscape is just another one of those golden ratio things where this pattern seems to fit all across nature. And that's definitely not a coincidence. Okay. Okay, we won't go too in depth here, but I guess this is more drawings of the embryo. Okay, so it looks like microtubules inside this embryo are arranged, not random. So they're arranged. Okay, so it looks like maybe microtubules are actually responsible for choosing exactly how cleavages are happening. Interesting, when an egg is inverted. So simply changing the rotation of the egg completely messes with it. So wow, that's interesting. Okay, so maybe the egg's able to detect its rotation based on where the fluid is, right? And maybe the fluid has to exactly be here, like on this lining. So the moment it sloshes somewhere else, it can't differentiate and grow. So wow, very interesting. Cortical rotation aligns microtubules via global torque. So I don't know what all that means, but I think the point is like there's fluid and these dots are the microtubules, and somehow these, when it's aligned perfectly like this, then it's able to move to the next step. So sloshing of fluid. Okay. So I mean, in summary here, I guess we're summarizing here. Memory depends on neural cytoskeleton. We talked about that, just microtubules. And obviously it's linked to all these things, plasticity, right? The long-term potentiation, more receptors, less receptors. Hallmark of neurodegenerative diseases, not only Alzheimer's, right? Just Parkinson's, Huntington's, stuff like that. And then that our cytoskeleton of microtubes are affected by anesthetics, right? They change, I think they bind to tryptophan or something. So again, implicated here. And because, in psychology, it's so hard to explain all the subjective things, realities, maybe quantum models, we can fit them on to explain all this psychology changes. So quantum coherence equals healthy state. So I think before it was referenced that when meditation leads to coherence, so maybe meditation is our healthy state. Decoherence is disease. And so maybe locating where the discoherence is kind of locating where the disease is. because cancer is a common example. So it's a common example because obviously like those cancer cells are discoherent. They do their own thing, right? They don't listen to the rest of the cells now. So, okay. Bioelectric medicine. So that's an EKG right there of the heart. That's a EEG. First one's, I think, electrocardiogram. Second one's electro, I don't know what EEG is actually. Electro something else gram then. Okay. Non-invasive signals, yes. Detect early stage. I think this is really more important than ever. I mean, if you guys know Elon Musk, he has that startup, I think it's called Neuralink. I think this is kind of exactly what he's doing. Although his is a bit more invasive, because I think some things have to be, not have to be invasive, but when you're more invasive, you get to go through the different layers of the brain, and different layers of the brain really deliver mean different things. Just a quick example, like our vision is processed at the back of our head in the occipital lobe, but different layers of the occipital lobe, like the deepest layer, represents different points of processing. So I think the outer layer is the most processed image, whereas the inner layer is the most raw one, right? So if we never invade, then we would never be able to get those distinct signals at the beginning, at the end, right? It's just like, I'm not even gonna make that analogy, that's too far-fetched, but the point is, invading the brain might give us more ideas about different levels of processing. But bioelectric medicine, I think these are questions, right? I think like there's literally a company out there now that's really interested in this. Okay, Michael Levin. Morphogenesis and cancer. The living matrix. Oh, another interesting example. So I guess here we're talking more that maybe our microtubules function as biological wires. So very interesting thought. And now we're taking it seriously now. So let's see. So what's the conductivity of a microtubule, right? Copper, very conductive, rubber, an insulator. So, okay. Bioelectronic future.
So there's 6 minutes left in the teaching session. You don't have to end right on time.
Okay. I mean, this is the absolute last slide right here. We can just read this last slide then.
Okay.
So we have no choice but to accept the world as it seems, but should be aware that the world as it seems is not the world as it is. As it seems is up to us, right? Our reality. But is that how the world is? We cannot know anything about the world as it is, other than that there is a world as it is. Wow, very deep. But it's true, like just envision alone, like you guys seen the electromagnetic spectrum, right? It's massive, but we only see a sliver, right? But even our little sliver gives us all these vibrant colors. So like, what are we missing out, right? There's so many other wavelengths that we don't get to see. Obviously all the colors made up, but the fact that we get to see such a sliver and still be able to interact and survive in this world, like just that sliver is enough to interact with the world. But there's so much other information in the world we physically can't capture, right? Which is extraordinary. I mean, yeah. So like sound as well, right? Sound only works if there's objects there, right? If there's no object, like in space, there's no such thing as sound. So sound is a very particular, interesting one. And then smell and taste, just crazy that we only get to, like our tools can only make us sense this much, but it's already overwhelming. Like it's already so much information that we can capture. So yeah, I think that's very interesting. Yeah, so I mean that really ends this presentation, but I guess I want to leave you guys with the thought of like, in 50 years and 70 years, how do you think our bodies will be like manipulated in such a way? Like for example, we have some people have Apple Watches, I don't, but some people have an Apple Watch and they can already constantly record EKGs, maybe not as accurately, but the point is you have data, right? You can record EKGs constantly. So that's one point of data that about our health, right? But then, like previously in the biometric medicine slide, right, we talk about getting more information, like the health of our body, like this Neuralink that there's implanted things in our brain, gathering more information. Maybe that's the future, right? We really become a little bit like cyborgs. What do you guys think on that?
Why don't we unshare and
Okay.
then you can see the students and do some interaction. There we go. Okay, so any questions? The exam questions are pretty simple things. So to some extent, how much you get into the details here, it's not simply motivated by your grade in the course. In other words, you can just sort of skim on this surface here. But I also think it makes you a much more interesting person if you do get into the details of some of this, right? So, I mean, the payoff might not be a higher grade in the course, but you're a much more memorable person at parties and stuff if you really get into the parts of this that interest you. And then maybe you'll get into it so far that you have to deal with the math that we're not dealing with at the moment. But yeah.
Yeah, I really want to ask you guys, like, do you think it's ethical to do the brain implants that Neuralink is doing? I really want to know your guys' thoughts on that and an ethical perspective. There's no laws right now talking about like brain implants, but what do you guys think about that? Like having that, it's really subconscious data, right? It's not even conscious anymore, right? But that data talks about our subconscious now. So what do you guys think about that?
I would try to answer, but I don't know much about Neuralink and whatever Elon Musk does, because honestly, like, I live in a cave, and I'm fine here. But this was such an interesting couple of lectures because, I've taken a couple of classes on quantum physics, and like, it's just so inconceivable to me how like quantum physics can be applied like biologically, because usually when you talk about like the classical experiments and stuff, it's all to do with like like quantum behavior and like gases and like superconductors and stuff and very like non-biological systems. So I'm just still wrapping my head around that.
Yeah, this is a great point. Are you like a physicist yourself, or?
I have a minor in physical sciences, so I've only dabbled.
Yeah, no, that's so interesting. That's so true, right? But I guess in some ways we think about if we dive deep into anything at the small scale is quantum. So maybe that's how it all connects, right? I don't know. I'm definitely not a quantum person myself, but yeah, good points.
So there's some interesting things from Thomas Banks in the chat. We should elect to do any procedure or operation they fully understand they're doing. Make the choice freely.
Yeah. Informed consent, absolutely.
Yeah.
That's another great point. I mean, ignore Neuralink, just like self-driving cars, right? Like there's no regulations for that right now. So that's true. Like policy is a bit slow when it comes to these innovations.
So, we had an expert in self-driving cars who was getting a PhD in self-driving car related stuff. And she gave a really excellent lecturer. I tried to interest her in what Elon Musk has said on videos about self-driving cars and other things. And she was so resistant to that. That was fascinating to me, that even that field is very, very diverse with you know, you can get a PhD in a fairly conservative area of self-driving cars, and your PhD advisor advises you not to pay any attention to, you know, Elon Musk. Meanwhile, we all know that Elon Musk is really helping to shape the future in a lot of ways.
Yeah.
So,
Oh, sorry. Go ahead. Go ahead.
oh, no, I was just saying we are a minute over now, but let's see. Yeah.
That's a great point that Anka brings up, right? Like, it's not even like fully informed consent ever, especially in this case. It's so innovative that you don't know the repercussions, right? That's a really good point. And then Thomas says that some people are just willing to take that reward. Yeah, I mean, you know, don't clinical trials normally take 10 years or something? I mean, COVID was like a 1-year clinical trial. So even though it's as informed as possible, it's not truly the best informed one could be. So
yeah, you guys may wonder what it's like to get vaccinated. So I was vaccinated, I guess last week Wednesday, and it was like the first day when they offered. It was so chaotic, but everything worked out fine for me. You know, I think even getting the website where you sign up to work was difficult if you were outside the Alberta Health Services network. And they told you to stay in your car until 5 minutes before, but then you go and you see these huge line of like over 100 people. You're thinking, well, aren't I better off being in that line than being in my car? You know, I mean, I'm sure it's less chaotic now, but there were, you know, official apologies about it. So.
congrats. How are your symptoms? Like, some people feel not so good after the vaccine.
Yeah, I had hardly any symptoms. I had some pain, which lasted 18 hours, but wasn't very severe, you know, just localized pain.
Yeah.
I didn't really have any other symptoms.
That's great.
I get my second dose on April the 3rd. So the course will still be active then. So you can get my report on the second dose. I guess there might be more symptoms in theory for the second dose. So we'll see what happens. Yeah. So next week is ethics from Michael Van Manen, and then Simon is back the week after. So that's the plan. Okay, well, I think that's another good teaching session. Thank you very much, Simon, and we'll see you back the week after next. So,
Thank you.
okay,
Thank you.
take care, everyone.
Bye-bye.
Bye.